Thank you, Katie. Good evening, everyone. My name is Amelia, and I am so excited to introduce you all to Yael S. Yael is very bright, and her passion for medicine and research is certainly very contagious, so you'll see that. Given her in-depth understanding of scientific concepts, we were able to learn a lot together in a very short period of time. And she was able to learn how to research her question and topic of interest. And you will see throughout her presentation how much she simplified all these concepts and showing in a model. So I am very proud of her, and I'm sure she has a lot of plans for the future. I wish we had more time to actually do experiments like real time or researching humans, but it takes a lot of time and approvals throughout the hospital. But anyways, I'm very proud of her, and I now invite her to take the floor.
Hi everyone, I'm Yael S. I'm in 12th grade. Next. So the question that my mentor and I decided to research was, is chronic psychological stress linked to epigenetic changes contributing to kidney disease? Next. So before I go into this very complex topic, I have to introduce you to my mentor, Amelia. Dr. Amelia Hollick is a physician and researcher. She currently manages the clinical research and gene therapy programs for the bone marrow transplant division at Cohen's Children's Medical Center. While she's extremely busy working as a doctor, researcher, lecturer, mother, and of course, magic mentor, she also enjoys reading and traveling. Next. So how did I even come across this topic? So when I interviewed for Magic, I expressed interest in learning about the process of conducting professional research and the stress disease connection. So we had to narrow it down to one specific idea. So I chose the connection between stress and the kidney. So initially, my mentor and I wanted to conduct research in a real lab on rats. We had a whole experiment plan, but due to age restrictions, we weren't able to go about that. So instead of being discouraged, my mentor decided to encourage me to conduct a systematic review. She explained to me that that would be something that I would have to be doing while I would be in the research field in the future. So after much discussion, we decided that the best way to answer my question, instead of illegally using the lab, would be to conduct a systematic review. Next. So what is a systematic review? The definition is a comprehensive summary of existing research on a specific topic in order to gain a clear and unbiased understanding of the topic in the scientific community. So first, my mentor's librarian, with the help of her librarian in Cohen's Children's Hospital, we were able to access dozens of databases and hundreds of research articles to find the ones that would be the most relevant for our topic. After accessing these studies, I had to look through every single one and see which ones would be the most relevant. After doing that, I summarized it into a spreadsheet, which is right here. And then after having it more organized, I had to assess the quality of each to see if not only it was relevant, but if the data was high quality enough to be included in a systematic review. So after this, I analyzed my findings, and I'm now in the process of compiling them into a single review for publication. So as you can see, this is an extremely lengthy process that can sometimes take years. And the fact that we were able to accomplish this much in so little time is really astounding. So I really thank Amelia for that. And I hope to publish my findings in the IJHSR High School Journal later this year. Next. So before I go into the results of my systematic review and the answer to our question, I have to explain epigenetics. So epigenetics explores how external factors and environmental influences can affect gene activity and expression. So these changes don't usually alter the sequence of DNA, but they can still cause potentially long-lasting effects on an organism. So one of the ways DNA can be changed in this way is through DNA methylation. DNA methylation is the process of adding a methyl group to the cytosine base of DNA, which blocks the protein that attaches to the DNA read molecule. This will turn that gene off. So some DNA molecules in the body must be methylated in order for harmful genes to be silenced. But if too many genes are methylated, then the genes that we need to conduct important functions throughout our bodies will be silenced, therefore leading to chronic disease. So one substance that can contribute to increased methylation are cytokines. So cytokines are immune cells that help control inflammation. There are several epigenetic factors that can lead to chronic inflammation and increased cytokines, including psychological stress, which I'm going to explain. So chronic stress will cause an increase in production in cytokines that are responsible for balancing methylation, leading to overmethylation and disease. Next. So the results of our experiment and our systematic review and the answer to our question are located in this model. So this model explains the process that the DNA goes through in the kidney when exposed to a stressor, ultimately leading to chronic disease. So first, the body is exposed to an epigenetic factor like environmental stress, which we all know can cause a person to neglect their health and make poor food choices. These food choices can cause hyperglycemia, which is high blood sugar, or hypertension, which is high blood pressure. As a person continues to be exposed to the stress, gene expression can be modified in a variety of ways, such as DNA methylation. So this occurs because the psychological stress can stimulate the inflammatory cytokine IL-6, which is an inflammatory protein involved in the regulation of DNA methyltransferase. DNA methyltransferase is an enzyme that's responsible for adding methyl groups to DNA molecules. This causes the DNA methyltransferase to add extra methyl groups to the DNA as it's being transcribed, therefore leading to epigenetic changes throughout the cells. So the methylation of genes that occurs due to chronic stress and inflammation silences genes that are essential for the health of the kidney and the person as a whole, which is directly linked to the decline of kidney function and renal fibrosis or end-stage renal failure. So through this review, we learned that patients with chronic kidney disease may have an increase in the amount of DNA methylation of the kidney as a consequence of psychological stress. Next. So I made a model, and it's not the best video, but so bear with me. So in order for you to get a closer look at the difference between the DNA of a stressed kidney and a normal kidney, which is that side, I created this model. So here you can see that on this side, we have the normal sugar phosphate backbone, which is present on all DNA. And if you look closer, then you have the DNA, you have the regular DNA base groups. So you have adenine and thymine and cytosine and guanine. And then we see here on the cytosine, we have these methyl groups on the diseased kidney. So we learned previously that as a result of inflammation, there is an increased cytokine response, which causes the cytokines to attach to the cytosine of the DNA because of the cytokine stimulating DNA methyltransferase to produce more methylation. This methylation can directly cause genes that are essential in kidney function to be shut down, leading to renal fibrosis and chronic kidney disease. So that's what this model is showing. Not the best video, but yeah. Next. So the highlights of magic, Next, please. So the highlight of magic for me has definitely been being able to have a mentor who has just such an incredible wealth of knowledge for me to learn from, especially in a field of interest. And I've really been able to discuss my future STEM aspirations with her, which has been really incredible for me. The challenges definitely were time management. We're both incredibly busy. Obviously, her work's probably a little more important than mine. So it was sometimes hard for us to coordinate with each other, but my mentor was incredibly flexible and we made it work. And I really did look forward to our meetings every single week. And I can't wait to be able to implement the knowledge that I learned from this in the future. Thank you.
Wonderful work, Yael. Does anyone have any questions?
Did the studies control for other risk factors that may have led to end-stage renal disease as opposed to psychological stress?
So he asked, did the studies control for any risk factors that could have led, that besides psychological stress, that could have led to end-stage renal disease? So we looked through a lot of studies. So some of them didn't end up being directly relevant to what we were studying, but that was kind of part of our whole process of conducting the systematic review, was to kind of weed out the ones that weren't as high quality, that didn't pertain to our subject as much.
Wonderful, thank you. Are there any other questions, either in the classroom or over Zoom?
What were you planning on doing if you could illegally get into the lab?
Oh, it's actually really cool. So basically we were gonna take rats and expose them to like a chronic stressor. It's kind of like you would, like you put them, I think containment is the way that you would induce psychological stress. And then you'd have to kill them. And then you'd have to extract the kidney and you would look under a microscope to see if there are any structural or changes to the DNA. But we weren't able to do that because I'm not 18, but next year or the year after.
I'm recruiting her next year.